So yeah, I'm really grateful to have the opportunity to facilitate a discussion on small biotech and interest in CMT. These are two of my favorite topics. As Edred said, I did a lot of research in CMT and in training as a physician scientist, and trained in neurology, and then joined Third Rock Ventures to sort of systematically create biotechs, and have really enjoyed learning about drug development, and eager to hear what our panelists have to say about what interested them in CMT, why they have chosen CMT for their programs, and some of the challenges that they've faced, and particularly trying to identify things that we can do as a community and as a foundation to help small biotech companies. So why don't we start with introductions, maybe starting at the end of the table, and tell us a little bit about your companies, the targets and indications that you've chosen.
Sure. Nice to be here. My name is Thomas Neenan. I'm one of the co-founders, and I'm the chief business officer of Nanite Pharmaceuticals. So Nanite's a 2-year-old company, more or less. We're based in Seaport here. We're really focused on developing non-viral and non-LNP vectors for gene therapy. So this is really sort of semi-blue sky, but we're huge believers that there's a enormous design space here that has not been explored, and that's what we're doing. So we're a group of mainly chemical engineers, material scientists, and so we just started about 2 years ago, and we got interested in CMT because I might met a couple of meetings ago. I had met with Clary and Susan, who are very, shall we say, persuavy. So they were quite invested in finding people who are willing to work on this problem, which is a big problem. How do you deliver things to Schwann cells? If you could crack that nut, it opens up an enormous vista of opportunity here. So that's how we got interested in CMT. The other reason for me is that one of my last companies was I've done a couple of companies based on iron overload, and in that world, I was interacting quite a bit with Cooley's Anemia Foundation and sickle cell groups. And so I've really learned the lesson that, what you really want to do here is engage with patient groups early and kind of understand what do they want, as opposed to, when you're a little company, you're always looking at big pharmas, that's your customer, but trying to stay focused on your customers, the patient here. And so what's really needed here? And I think in our case, we really believe that methods to effectively deliver viral or deliver gene therapies to Schwann cells would be a huge breakthrough. So I'll stop there.
Great, thanks. Hi, Steve O'Connor. I'm the CEO of Shift Pharmaceuticals. I co-founded Shift a few years ago with Chris Lorson. Chris, you can raise your hand. Most of the people in the room know Chris. We were founded based on targets for spinal muscular atrophy, and we still have those programs going forward for adult patients with SMA using ASOs. And a couple years ago, Chris met Susan at a conference, and she shoved the Ionis paper in his face and said, "You guys should do this too." And we actually got started with that because we were really confident we could take what we had done previously and affect it in that, and have gotten in this sense because of, of course, what everyone says, the great need, et cetera. But one of the things I always do as an entrepreneur, I've started about 15 companies over the years, is make sure before I even get into anything is, can I technically do it? And so we felt pretty confident that our technology could apply, which was, then we looked at the market capability and the patient need, et cetera, et cetera. But I don't ever want to work on anything if I don't think I can do it. So anyway, we're glad we're here. Thanks.
Hi, everyone. I'm Masha Zečević. I am an incoming chief business officer at Samsara Therapeutics. Samsara Therapeutics is a preclinical stage company that is based in Europe and in the US. Some of us are here in Boston. Others, our research facilities are in Oxford, and we have a team in Berlin as well. The company is founded around novel and proprietary technology to find new molecules that facilitate and increase autophagy itself. This is a very established way in which cells get rid of unwanted, undesired proteins. And we've been applying this technology to find new molecules that act on different diseases, mainly in neurology, that share this common issue of dealing with accumulation of unwanted protein. And some subtypes of CMT certainly fall within this field. And from everything that we have seen so far in animal data and in cell data, it looks like we have very interesting novel chemical matter that could be applied for CMT1A. Now, full disclosure, I am new to this therapeutic area. CMT is new to me. I come from a different background in terms of working on a rare disease very closely with patient community and foundations, and that is cystic fibrosis. Well, during my time at another company where we discovered molecules and took them into clinical trial up all the way until phase 2, interactions with patients, with research foundations, with KOLs have been absolutely instrumental and pivotal for us to get us from an idea with early grants all the way to phase 2 studies. And I'm hoping that the lessons learned, which were all positive, can be applied to CMT as well at Samsara Therapeutics.
Hello, I'm Douglas Fowler. I'm the Chief Medical Officer for Oryzon Genomics. Oryzon is a company focused on epigenetic therapies. Probably most of you know this, but epigenetics is a layer of gene control on top of the sequence of the genes. And we use small molecules to regulate gene expression using the epigenetic control system. Our interest in CMT is because we've developed a highly specific, highly active HDAC6 inhibitor as part of our epigenetic therapeutic profile. I know some people out there are already saying, but HDAC6 is not an epigenetic agent. All of the other HDACs are epigenetic modulators, and it turns out HDAC6 is not. It has a very different function, but a member of the same family. Nonetheless, we have a molecule that we're interested in potentially using to help patients with CMT.
Thanks.
Hi, I'm Brian Lafitte, Chief Scientific Officer for DTX Pharma, now an employee of Novartis. I'll let Artie tell you the story about how we got into CMT, because that's really his story, but I'll say that from our point of view, we were a small company with a technology that was good at delivery of siRNA to extrahepatic locations. So we looked for disorders which fit that bill, meaning that things which had an overexpression or an extra copy made perfect sense for us. So we principally looked at genetic gain of function disorders. And of course, we looked for diseases with strong pathophysiology. We always talk about having good animal models so we can prove our compounds work, and then scoring systems in the clinic. Many things that you've heard about today that are being developed for CMT, all CMT subtypes are things that we look for in deciding which disease areas we would focus on.
Hi, my name is Sylvain Celanire. I'm CEO of Augustine Therapeutics. So Augustine Therapeutics is a biotech company, spin-off of the VIB K11 Institute in Belgium. We have been creating Augustine Therapeutics from the work of Ludo van den Bos, who has been the first to discover the role of HDAC6, same target, in CMT 10 years ago, that led to the creation of Augustine. So we have been developing drugs very focused on this target class. We are extending that to peripheral neuropathy, CIPN, and ALS more recently as well. So we have a pipeline of 2 lines, one peripheral, one central for different indication. And we came to the CMT field because of the work of Ludo very early on. And the goal is to apply that to all subtype of CMT. We have data on CMT2A, CMT2F, CMT2D, and CMT1A. And so we plan to apply that to all the patients we can.
I'll keep this brief so we can get to the discussion, but I'm already an unemployed scientist, founder of DTX Pharma. We'll talk, more about our experiences raising capital and such as we go along. As for what I'm doing now, I did promise Cleary and Pat and Susan that I wouldn't retire until we had a treatment. So my work is not done, so I'm going to work with them on the sidelines to make sure there's 3 or 4 more DTXs over the coming years.
Thanks. Okay, so I think I'd like to try to touch on, I think from the day, everyone probably has a sense of what excites everyone about CMT, and you guys alluded to that. Just a tremendous amount of unmet need, no approved therapies. I think talked a little bit about this, but what do the panelists find most daunting about CMT as a therapeutic area for drug development, and what's missing that is, you know, something that we could try to, a gap we could try to close as a community? Go ahead.
Well, I'd like to be very pragmatic and say that what worries me the most is the lack of a clear pathway to approval. And this is a really big deal. To me, it's impossible to design a good clinical development pathway unless you know what your final trial is going to look like, unless you know what mark you have to hit. And do we have a gold standard for CMT for approval? I don't think so. We know what's been used. We know that a number of phase 3 endpoints haven't worked. That doesn't mean they're not the right endpoint. It may mean that the drug didn't work, but we don't know. The current trial, the current phase 3 trial, is using the yes, thank you. The overall neuropath test. And is that the right test? The FDA has not said no to it, but they haven't said yes to it. The trial hasn't read out. Is it the right test for CMT? It wasn't designed for CMT. Is it a test designed for a clinical trial as opposed to working in the clinic? And those are very different characteristics that, as far as trials go. Is it the right trial for all the right endpoint for all CMT? We don't know. This makes it hard. And, you know, the first question many investors ask is, what's your pathway to approval? What's your endpoint in your phase 3 trial? What has the FDA said about it? So this is very difficult, and it makes it, maybe we can talk later about some ways to try to solve this, but it makes drug development very difficult. And, you know, for other diseases, it's much simpler. A hit in this with a given endpoint and approval will help clear up things a little bit, but it still won't mean it's the right endpoint for your trial. So for me, this is what patients want. They want an endpoint that will give them an approved drug. We need that endpoint. As was said earlier today, and very appropriately, we also need to make sure that endpoint, whatever it is, you know, a shift in a scale really reflects patient benefit. How much does it have to shift to reflect patient benefit? Does it reflect patient benefit? Does this scale, which is usually made to measure decreases in function, will it measure increases in function? So to me, that's the most daunting thing about development. Everything else is easy.
That's true. And I think that highlights what the first approval would represent for the community. Any other comments on what's daunting?
I mean, just maybe to build on that, which I absolutely fully agree. In the uncertainty of a regulatory pathway, what do we look for? We look for some biomarkers. We look for some signals that we can capture in the interim as we transition from preclinical into early clinical and clinical development. We look for those reassuring signals that we are on the right path, and then hopefully the clinically meaningful signal will eventually accrue and appear in further studies. So maybe a call to the research community and to the patient communities to really think about describing the patients who are by definition heterogeneous, both in their molecular markers as well as in their presentation, with additional elements that we might borrow and use and implement in our clinical trials. Maybe validate new molecular markers, correlate them with specific genotypes, correlate them with certain pathologies, with certain presentations and severities, and help us find tools that we can continue to explore and validate in our clinical trials. And maybe eventually we might be able to have second and third generation of drugs that will be able to be approved on the basis of something simpler, a simpler, shorter signal that we can capture, and therefore accelerate the overall R&D engine for this therapeutic area.
I'm Steve O'Connor. We're developing a drug for CMT1A, and there's been a number of great clinical studies on natural history progression of that recently, and some of the authors are in this room. And I'm going to mess up the acronym, and I think it's the RSLM or something metric. Can one of the authors tell me if I'm messing up that acronym? It's basically physical functionality of CMT1A patients. How do they walk? How can they use their arms? How's their balance, et cetera? And it shows with excellent p-values in 2 years, clear progression. And we're going to try to make people walk better and with our drug. And so, and I think the clinicians, most of the clinicians we've talked to agree that that will be a great clinical outcome measurement that everyone who's a research clinical neurologist will probably accept as the test to do for physical outcome. So my bigger fear is I'm having kind of biomarkers forced down my throat by some people. And it's not really clear other than the fat fraction, which is technically not really a biomarker, which is there's some studies showing that fat fraction through MRI measurement is progressing very quickly. It's not clear to me yet what biomarkers, and listen, DTX is going towards the clinic, right? And you guys might have one, I'm sure you can't tell us, but our goal is to make people function better. And so we want our primary outcome to be physical behavior. And from what I've seen, there is good natural history on that already. And there'll probably be more, so.
So I gotta press the button. So I feel like Brian and I could probably give like a 6-hour seminar on all the challenges we encountered over the last couple of years, but a couple of, and they're probably different for CMT1A versus the other CMTs. And so I think with respect to, CMT2 and some of the other ones, I think one of the challenges there is they're very rare. And oftentimes you kind of need to remove a gene and then replace a gene. And so, getting investor traction, interest in those is going to be, is challenging and daunting. In 1A specifically, and you guys can stop me because I could go on forever about this, threading the needle, there's another disorder called HNPP, which is, over or not having enough PMP22. And, there's a real risk when you give a 1A treatment that you would induce that phenotype, and how that presents in, an adult with maybe not as plastic of a nervous system, we don't know. So that scares me. I think, we don't know what time to intervene. I think that's really important. It's probably true for all of these. And when you're doing clinical development, you have investors involved, you don't get the timing right, you might not get another shot. So I have like 5 more things written here, but I'll pass on going through them all. But certainly, even though there's a lot of positives, it's not easy to develop a drug in this space.
Might be a good point to transition to talking a little about what you hear from investors. And I know we're just preceding a panel with a group of really thoughtful investors, but I'm sure also not all of the investors you've talked to have been as informed about CMT. And so how have investors reacted to interest in CMT? What are their big questions or unknowns? And what do you wish they knew before you pitch them on programs in CMT?
And start. We are fundraising now. We are in a Series A fundraising. We have been a first seed round in 2019 when we created the company with European investors mainly. So Belgium-based and French-Belgium investors. And we started with the CMT, which as a primary indication. After 3 years of work in the lab, advancing the pipeline, we are now at a good state where we can raise money to go to the clinic, which is the aim of next year, to entering first in man. And so we have a good time to now raise the money we need to go there, even in a phase 1b/2a in patient. And so the first time when we reached out with the first set of investors, definitely CMT was not the most great idea from their side due to the complexity of the clinical development, the lack of endpoint, also the lack of clinical trial, successful one. And at the same time, Pharnex was developing their first phase 3 and to redo a second phase 3 trial that we aim to, I would say, I guess we have some data by the end of the year. So there was a lot of enthusiasm from us, but a lot of skepticism from the investors at the time. We'll see what's come, but we need a success somewhere. Whatever I would say competitors in the field, if there's a success, there's a way to do better also as well. And so CMT was not the first really indication that we say the investment community see for health tactics. Initially, there's a lot of plenty of indication where health tactics can play a role. And we focus really on how we can advance this target class with new chemistry, something completely different from the other third generation of compounds. And so we build a case for CMT, but also we build a case for other indication to develop CMT. The time we get all the clinical endpoints, the biomarkers, should it be MRI, should it be NFL, a lot of, I would say, things has happened with tofersen approval with NFL based on NFL only. And that gives hope that a surrogate biomarker can really make a change and reassure the investors. And that was what we play into advancing the case. After 100 meeting with investors for the past year, we get to the closing. So very exciting. We have US big pharma investors participating, US investors, European investors, and we build the case because we build the pipeline for that, and not only one side indication. And beyond CMT, there's neuropathy, there's ALS, there's a lot of, I would say, common, I would say, a thing that we can do with HDAC6 inhibitors. But the investors see the trends and also see the potential beyond CMT1A, which is the first subtype we are talking to. And thanks to the CMT Research Foundation, where we get funding support, scientific support as well, we create something, I would say, meaningful for the investors, that there's some from US, from the Europe community. There's, I would say, logics in the fundraising perspective. So
okay.
We are the right
track. Yeah, congrats on the A. Yeah. So I heard from that actually the tofersen approval and understanding neurofilament as a qualified biomarker of motor neuron degeneration, you think was really important in catalyzing investor interest.
What we done from the first time we do the studies, we include NFL from the start in preclinical model to translate the best for the patient later on, because it will be a potential outcome measure and biomarker. And we plug all biomarker we can in the mice model of CMT1A or using iPSCs as well. There's a great chance to get some target engagement as well. So we need to build for the investors really the case that we can de-risk if there's a lack of clinical outcome measure.
Mm-hmm. Thanks.
Yeah, if I could just re-echo that. I mean, what Sylvain said. I mean, I think it shouldn't be a surprise to anybody that most investors look at CMT as a tough nut for all the reasons we've talked about today. It's heterogeneous, it's slow progressing, which is the best news a patient can hear, but it's not a great fundraising pitch. That's a very slow progressing disease. So, you know, our position has been that CMT, you know, we're a platform company, so we're trying to develop technologies that allow us to deliver to tissues that other people can't deliver to. So our pitch has been that if we can deliver to Schwann cells, we can start to deliver to other tough tissues. And also, if you can deliver the peripheral nervous system, there's lots of other opportunities within the peripheral nervous system. But I think it is interesting to hear what the VC panel says next. But I would say this is not a trivial area if that's what you're going out with, you know, as your primary pitch. And in our case, it's part of a portfolio of opportunity that we see.
Yeah, I think that's definitely true. I think time to clinical proof of concept is so important for a small biotech, and that's fairly long in CMT. And I think a lot of what you're hearing is what are ways that we can shorten that and reduce the patient numbers we'd need to achieve clinical proof of concept, whether it be a qualified biomarker which we don't have. Like, and what do we think the first qualified biomarker will be, and what will that qualification process look like? Yeah.
Just very quickly, sorry to interrupt. The virtual audience is having a hard time hearing, so if we could just
Can you
speak
hear
up
the
a little.
speakers?
I think you specifically.
Okay, sorry.
Yeah, sorry.
Might be a little loud for the people in the room after this. I'd like to talk a little bit about what you guys hear from the regulators, and whether you think I know you commented on regulators' reaction to ALS, but what have we heard specifically from regulators on CMT? And is that encouraging, or is it challenging? And how do you think, or what do you think it'll take for regulators to acknowledge how hard this could be and create a path?
I think it will be hard for Brian and I in particular to talk about what we heard from regulators, but maybe on your last question, I think probably the most important thing that I think you're asking, what could we do to create a path? And I think probably the most important thing is, CMTRF had a listening session, them being exposed to patients. This is a fairly new disease to whether it's investors or the FDA. And so I think getting in front of the FDA, saying what, in that panel this morning, there's issues with breathing, you're falling all the time, these things, I think we have to get in front of the FDA and get in their face about how devastating of a disease this really is. And so I can't comment on what regulators have said to us, but I do think that will be really important going forward. And they will, take that information and use it to guide development of whether it's Shift or Novartis or some of the other companies.
Can I just expand on that, because there are remarkable things foundations can do. I know that you've met with the FDA. I'm on the board of a foundation for another rare disease, and this tiny foundation set up a large meeting with the FDA, and I mean hundreds of people, and they invited public, patients, industry, academics, and the FDA. And leadership of the FDA showed up. It was an excellent meeting. There was the FDA agreed that they had learned a lot from this, not the science, but hearing from patients, what really bothers them, to let them think more about what kind of endpoints are important. But to me, the most interesting thing was the follow-up. This little foundation didn't stop there. They said, let's have another meeting. And the next meeting, we want to talk about endpoints. And we want to discuss endpoints with you, clinical endpoints for registration. The FDA agreed. We ended up setting up committees, and we had a committee for cardiac endpoints, a committee for neuropathy endpoints, and a committee for hepatic endpoints, because this disease affects all of those systems. And different parts of the FDA, who sometimes the disease is divided into different parts in the FDA, all have participated, all have come up with recommendations. We've written a white paper on what the endpoint should be in these trials. And this is the kind of thing only a foundation can do. Most of you know that when a company comes in, it's not a nice talk back and forth generally. Sometimes it is, and in rare diseases it can be. But there are two positions, people have two positions going in. It's not a bargaining setup. And here, this was a honestly brokered set of meetings, and they're still going on, because there are even other endpoints that patients have said, these are very important to us, and we're trying to set up registrational criteria based on improvement in those endpoints. So it can, endpoints can be addressed, trial designs can be addressed, agreements can be made with the FDA, all done by foundations.
Yeah, it's definitely true. I think it's a privileged position that foundations have to bring people together across different types of organizations and try to get things done. Any other comments on the regulatory piece? Okay, want to ask, so we've talked about a few things that are really important, but are a little less tangible than maybe thinking about what experiments or what organizations and people you bring together and what problem you tried to solve. So if you had, if there were sort of, you imagine a world where there were no red tape and limitless funds, what experiment would you try to fund that you think would catalyze drug development in CMT for all types of, and benefit all of the companies on the panel? And I think of, fields like the cancer that have really, the Cancer Genome Atlas, which has just been a remarkable resource for all companies that are working in the field. What are the resources that the community could generate that are like that? Unbiased data sets. Help me think of things that we could really try to focus on at the foundation, try to catalyze.
So that's not exactly what your email said. You said I would get infinite amount of money, and so now you're making it much more cautious. So I will go away from the infinite amount of money because I know I don't get that. For someone like me, and again, I have a PhD, but I have not been an academic, so I don't worry about publishing too much or whatever. But access to raw data, like with these natural history studies, like I get the synopsis of snapshots of data where it was just done for enough, frankly, to publish a paper. And I want like every single piece of raw data when I'm actually giving a drug to an animal and watching that animal, right? I'm not trying to necessarily just get good enough statistics to have great standard deviations and p-values. Like now we do that too, right? But and I know this is a very non-academic thing, but if people could start getting like raw data, including bad data, and like one of the things that surprised us with our animals, and when Arthur Burgess said this morning, we do breed our own animals, by the way, we get them from Jackson Labs, but we keep track of the mommies and daddies in every single breed, okay? And we see them act differently. Children of the same animal do physically behave different, even if their gene is the same. And that's the same with patients, by the way, right? Patients that have a similar genetic or identical genetic profile will progress at a different age often in this disease. So we don't think we're seeing anything different. And we kind of had to learn those numbers on our own because all we saw in publications and from other groups was average and standard deviations. So I know this goes crazy against academics in general, but I think repositories of actual raw data of patient data that's genericized in animal data, that's natural history numbers, et cetera, would be really beneficial for me, I think, in general. Because, and I've experienced this in other industries and et cetera, which is oftentimes people like me have to recreate some of this, figure some of this stuff out again on our own when I think someone else already knew it. So.
If I can maybe borrow from my experience in cystic fibrosis. Cystic fibrosis is, for those who are not in that area, is sort of a prototype of a simple genetic disease. The gene was sequenced in the 80s, mutations were identified, patients were promised therapy just around the corner, given that now we know what the gene is and how to work. And then fast forward 30 years later, and new therapies were only starting to become available. But the field has completely changed, and what used to be a deadly disease now, thanks to prenatal screening and therapeutic options, patients can expect almost a full lifespan. Now, although the therapies are available, again, and although patients share the same mutations, oftentimes they have a different outcome. They have a different response to therapy, and they have a different disease course, and we do not understand why. In Europe specifically, these problem compounded with the lack of access to therapies because of the price, has driven the research community to come up with something new, something that didn't exist before. And that was a way to predict how an individual would react to a drug based on how their cells would react in a dish to this drug, and also predict and correlate how this cell behaves in a dish to what is the severity of the disease. And so this so-called the organoid approach triggered a momentum in a collection of samples from patients across many different centers, first in Europe and now in the US, with these repositories of hundreds of patient-derived organoids that are available to be studied. So we don't really need to fully understand what is happening on the cellular level. We know that this comes from a patient who has a certain disease and a certain mutation, but it allows us access to biological samples that we can study for a response to drugs, that we can study for profiling the patients, for finding out who might be a responder to a new therapy that is going into the clinic. So it allows us to do clinical trials in dishes before we actually go into clinical studies. But this was a combination between advancement in science and a bit of desperation due to the lack of access to approved therapies because of price that created this. And I imagine if we started creating and populating and maintaining and enriching these sort of databases with information about natural histories and patients, with all of these things that matter to them, the way they were describing it this morning in terms of falling and hearing loss and all these other things that might not necessarily be obvious endpoints, but really matter to them, we as a community might have access to to avatars of our patients that we could study over years to come, and hopefully advance the field even further with making it simpler for all of us to sort of pre-screen patients and drugs before we move into the clinic. So that was an extremely positive experience for us. We managed to test our drugs in 500 patients worth of organoids, and to find out pockets of those that we predicted would be responsive to our drugs before we extended the invitation for them to enroll into a clinical study.
So Will, your original email did say there was limitless funding here. So I think we've talked about this, all of these topics all day now. But I think the fundamental lessons and what you need here are pretty not straightforward, but I think Sharon Hesterly sort of went through this. So you want to identify the genes that cause the disease. You need to develop an animal model that recapitulates the disease. You need to find biomarkers that will allow you to do clinical trials. And probably what's missing maybe here in CMT is finding ways to identify patients, particularly slower progressing patients, much sooner so that that patient population for clinical trials can be identified. So it seems to me that in CMT, everything that should be done and can be is being done. It's just, could you do it if you had a lot more limitless money, as you were offering us this morning? You could do a lot more of it a lot more quickly. But I don't see that, the course of how CMT is going to be treated, it would seem we're on the right track here, right? So these pieces are being slotted in. Maybe there's gaps, and there's significant gaps, certainly on the animal models and perhaps on the biomarkers, but I think with limitless resources, we just do more of what's being done right now. So I think it's an optimistic scenario. It's just, if you're a patient, it's not just not moving fast enough.
So standardized publication and more broad publication of animal model natural history and sort of expectations around how therapy should be tested against each other, biorepositories, and the potential for biorepositories to offer ways to identify patients most likely to respond to a therapy and patient selection biomarkers, and then fill in gaps like other biomarkers, clinical outcome biomarkers. Anything from the audience for this exceptional panel of biotech leaders?
Well, if you had the infinite funding, you could accelerate development, right? You could do so many things because because of lack of funding, sometimes we cannot do long-term talks or anything. That's where I think getting infinite funding to accelerate clinical development, and because you're in a rare disease area, you have, not being too optimistic, but many regulatory advantages to accelerate that development, and then design your clinical development plan in order to optimize the development. So getting infinite funding does help in that process.
I forgot one more thing I'd like. I'd like a mouse model that only has one copy of human PMP22 in it. For side effects tox studies.
Yeah, actually, I want to follow up on what Dr. Zekovic said in that, she's talking about phenotypic models, right? I mean, all drug discovery used to be done in phenotypic models, right? Until you got the whole molecular biology revolution and everything went to be target-based. Okay, everything had to be molecular biomarkers, okay? And that's how drug discovery was done, okay? And the problem is it works with big populations and big diseases and things. It doesn't work with rare diseases because you can't get enough information because your population isn't large enough. So this is one of the reasons I think going back to phenotypic models, okay, for rare diseases, you don't have to completely describe the disease. You just have to have a disease state, then have your normal state. And as long as you get that, a functional aspect of that, okay, back up to near the wild type, the FDA is saying it will accept those, okay? And because you can't run it like a huge clinical trial because you don't have enough patients, you don't have enough money, okay? And so I think getting back to the phenotypic models, which again, before 1950, everything was phenotypic models. Lots of drugs were approved based upon those phenotypic models, okay? So I think the molecular biology, revolution was awesome, but I think it sometimes went too far, okay, where people only think that way. But there's different ways of doing drug discovery as well.
I think the principles of good target validation and discovery are the same for independent of the prevalence of the disease, though. And I think, if you look at the success in SMA and CF, it's because we knew the target, and there was human genetic validation of disease modification.
Right, but I agree with that. When you can get a target and you have enough money behind it, okay, you can still do it that way. If you don't have enough money and you have a wide variety of different targets, SMA was pretty clear that's what your target was. You didn't have very many variations of it, okay? Whereas here you're looking at, so many different variations of CMT, okay? And you can't put that amount of money and that effort on all of them. Just like with cystic fibrosis, you've got well over 200, 300 different variations that you're aware of. You can't do it, and they didn't. So they had to go to the organoids, okay, which is a phenotypic model.
Any other questions from the audience? Offer something?
Well, I guess I'm not necessarily in response to that, but in general, I've sort of sensed as we've been going, we've been a little bit pessimistic about drug development. I kind of just want to add a few points about how attractive a space CMT is for drug development, so that we leave here a little bit excited that you can actually raise money, you can do a clinical path. And so, I eat, live, breathe this pitch all the time, but I think we should remind ourselves, at least with CMT1A, there's 75,000 patients with a disease. By the way, that's more than DM1, DMD, and FSHD combined. There are at least 10 therapies in development, probably clinical development for some of them right now. And we don't even have one. Well, we actually have one in phase 3 for CMT1A. 2, we are being very pessimistic about the clinical path, but the reality is we can measure target engagement in patients. So that's a huge win. That's not approvable, but we know if our drug is doing what it's supposed to be doing. We can do electrophysiology in the clinic, right? And all the rodent studies, I don't know if it's going to translate it. We have no clue, right? Brian and I talk about this all the time, but we have that. you have decades of natural history data. Sorry, you can stop me if this
No,
is
it's
annoying. Decades of natural history data and more coming every day. And I bet you, Steve, if you ask the investigators generating the data, they will give it to you in some form. So, and then of course, from an investment perspective, the market is like maybe $4 to $8 billion market with competition at charging reasonable rates. So, I don't know. I'm trying to share with you some things that worked to raise money in the space. And make no mistake about it, 99 out of 100 people said no to DTX along the way. Even this year, Matt, an enlightened institutional investor, he can tell you that we weren't sure we were going to be able to raise money. And, of course, Ari would have stepped up hopefully, but, in January 1st of this year. And so, and maybe the other plug I'll just make is that institutional investors are not bad. They're supportive. They allow you to go faster. They allow you to stack risk. In my experiences, they're very reasonable. So different than some of the talk before. But in any case, I just thought it was important to mention that there's no reason why, like over the next 3 years, there shouldn't be 3 or 4 companies in CMT1A and more companies in the other CMTs, because there's a lot that has been done way before DTX or any of us came to the scene that we can ride the coattails of. So I just didn't want us to leave super pessimistic about it.
And I wanted to build on that, because this goes both for talking to investors, but also talking to business development partnerships, et cetera. Yeah, I agree with Artie. There's a couple other things he didn't mention. Anyone in the rare disease space has to be so grateful for the success of Spinraza. Because I don't know if any of you know, and I know this is CMT crowd, no one expected that drug to do as well as it did in the marketplace as it did. I mean, it went to $1 billion in sales in like a year in an even more rare disease. And that really got the entire industry more open and investors to rare disease drugs. So that was actually unexpected how well it did. And that development team was just remarkable, because remember, that was an intrathecal injection in infants. So even the idea of conducting that trial initially was very like, wow, because I know some of the players at Biogen and Ionis in the early days that were part of that. So we all have to be very thankful that for whatever reason that product is done so remarkably well and save people's lives for all the rare disease communities. And yeah, I'm very excited about CMT. I think when we first looked at it, a lot of those natural history studies had not been published yet. And so I would say one of your questions was, what do investors worry about? I mean, this is a non-deadly disease that people are really worried the clinical trials are going to take forever. That is really the biggest thing. All right. I don't think any partner investor that I've talked to worries about scientifically, can you do it somehow, right? I don't think anybody worries too much, other than any other drug, about, oh, are you going to kill people and get a good tox profile, et cetera. The biggest thing is, oh, it's a rare disease. It doesn't kill people normally. And I'm worried it's going to take you 10 years to do a clinical trial, in which case I'm not giving you any money whatsoever. I will say investors and partners view CMT1 as a different disease than the rest of CMT, because CMT1 by itself is a big enough market. So even though scientifically we aggregate them in some ways, CMT1 is kind of looked at as a disease on its own from an investor and potential partnership. So the whole aggregation of different things is not as big a concern for me with a CMT1A drug as it might be for someone developing one of the rare forms. So
I have a question for the panel. And Artie and Brian, you're not allowed to answer this question, but everybody else, I mean, it's been 2 months since Novartis announced the acquisition of DTX. And I know, Sylvain, you're out there raising money, and all of you are. Have you seen a change in your conversations with your investors since this announcement? Have you seen more interest, or people calling you now that you went to before and said no? Or is it too soon to tell?
No. Yes, the top 5 big pharma came to us because it was the first big pharma to come to the CMT field. And so we have due diligence from big pharma now. We have corporate venture more and more to the field that know neuromuscular disease that have been commercially successful in doing CNS, PNS, neuropathy. So let's make a change. We need to think that actually and finger crossed for the fundraising. But we see a change, and we know Bob Ballo. Bob has been working with Ludovan and Voss for many years as well. And the ALS field is also very, I think, helping in that kind of things. So no, we see a change. 1 year ago, the CMT was not really the good indication for us, and finally, this is where now the big pharma and corporate venture came. So let's make a change, yeah, sure.
Yeah, Susan, I think that's definitely true. I think the acquisition has definitely piqued a lot of people's interest. So I would say, you know, over the last couple of months, I think it's much easier to get people to talk about CMT. So I think
Well, you know, I'll help.
that.
A question.
And I thought with the interest we were receiving was because of our data. We made.
So you just mentioned that the industry sees CMT1A as a entity of its own. Is it because it shares some of its pathology with other neuropathies such as chemo-induced or diabetic-induced neuropathies? Is there some overlap there in their view?
Yeah.
No, just because it's a large patient population where a single drug could probably go affect all of those same patients with a very clear molecular mode of action. And so I'm not, by the way, I'm not saying scientists in industry look at it this way. I'm talking about more as a business opportunity. It's looked as, oh, that's a business opportunity. Same mode of action, same patient group, wow, these other 20, 30, 40, 50, you know what I mean? Oh, those are 20, 30, 40 different business opportunities is kind of what I meant, not scientifically.
Thank you.
I would say yes. I would say that sharing, I would say, axonal damage or axonopathy in multiple indication give a chance that you can de-risk your strategy. And so we see synergies with the biomarkers, with the target engagement from one disease to another. We know that CIPN has some link with CMT for some patients. So then some people have discovered having CMT because they had the CIPN before. So we see some synergy between that and that you can extend this therapy to other neuropathy. That also brings the opportunity even bigger, at least for pharma.
Great. And I think one thing that Artie didn't touch on is that makes CMT great is the community of researchers that are all extremely collaborative and eager to talk about CMT, and also the patients and patient advocacy groups that and we need so much more interest. So hopefully, I think you can walk 5 minutes that way and throw a rock and hit 10 biotech companies, and hopefully in a few years we can say we can hit 10 biotech companies working on CMT. So thanks.